Hey guys.
Hi, everybody.
Hello.
Hello. Hello, everyone.
Jake, did you end up going to Berlin?
No, I'm going to go in a couple weeks.
Nice. I didn't expect the office background, so.
Yeah. So we are live. Hello, everyone.
Hi, everyone.
Hey.
Hey everyone. Are we expecting anyone else to join, or is it just the five of us?
I think we can go. Yes.
Okay. Hey everyone. We are really excited today to walk through some examples and just discuss BIOS, the AI scientist, or as Jake likes to say, AI for scientists. Jake, you've obviously been playing around a lot with BIOS. I know that you had some early access to it. Would really love if we can, dive into some of the details. If you have any, example conversations that you have that are like really interesting, just to show the anyone that's watching this what they might expect from this and like where you see the potential of this type of product, this type of application going.
Yeah, I love it. I've had a lot of fun playing with BIOS. I guess maybe I'll start with a little teaser for the audience. I did sort of two versions of the prompt "Make me rich" in BIOS. I tried sort of two different strategies of give me a technical research plan that is going to make me a lot of money, and I'll sort of share the results of those. I'm not rich yet, but we'll see how those go. So that was my I'll share two different versions of that kind of strategy. Maybe I should give people a little background on myself so they can understand like where I'm coming from, what my particular use case is in BIOS, and what I'm looking for in an AI scientist. So I'm a scientist by training. I have a PhD in systems biology from Harvard Medical School. My research focused on metabolic engineering. So basically genetically modifying microbes to do interesting things or to make useful products. I was an academic for a long time. I was a professor in Paris, and then a few years ago, I went to work for a genetic engineering company called Ginkgo Bioworks. And Ginkgo Bioworks was a platform company that sells genetic engineering as a service. So people would come to Ginkgo because they wanted a microbe that could make something. Ginkgo does the genetic engineering that's required to get that microbe to make something valuable, and licenses it to the customer, and the customer uses it to make their product. And my particular role in Ginkgo was called developer evangelist. It was kind of a technical communications role, and it was about translating between what Ginkgo could do, what we could do with genetic engineering, the kind of problems that genetic engineering can solve, and the kind of problems that customers have. So we work as scientists in industry with people who are not scientists and who have sort of problems that they need solved, like in the industrial production space, and they don't really care about the sort like the nuances of the very beautiful genetic designs that you use to solve their problems. They just want to know what biology can do for them, and what it can get, and if it's going to improve their bottom line. And so it was in that experience in industry that I became a kind of a product person, and I became interested in not as much like what is the beautiful novel elegant frontier science that we can do with genetic engineering, but how do we actually map that capability to something that a customer wants, to something that solves a problem in the real world? And so this is my interest in BIOS. This is what I'm very, very curious about in the space of AI scientists. So I think like the first question is like, who are AI scientists for? Right? Like who is the customer of an AI scientist? Whose problems are AI scientists solving? I hope that the answer is scientists like me. I hope that these AI scientists are targeted to be tools that are useful to people like me. That's the sort of assumption that I'm using when I go in to work with BIOS. But I'm also interested in a few other use cases. I'm interested particularly in the power of these kind of models to translate between the needs of a non-specialist, someone who has a particular use case, doesn't have a PhD in genetic engineering, but knows enough about the field to realize that there might be a product that they can create or a problem that genetic engineering can solve for them. And a tool like BIOS can mediate that kind of an interaction and get them to a place where they have something that is scientifically rigorous, scientifically plausible, without needing the sort of like years of training. The thing that is the sort of most exciting to me about that is that by democratizing access to that tool, you're going to get a lot more different takes on problems that biology needs to solve, problems that genetic engineering needs to solve. I think a real bottleneck that we've had in genetic engineering in the course of my career is that too much of it is done like by scientists for scientists. It's we sort of create toy models and we publish them in journals, and they don't really solve any problems for anyone. And we only really talk to ourselves. And so the power of a tool like BIOS to bring people into genetic engineering and say, like, no, I don't actually, like, I don't care if this is going to publish in Science Magazine. I care if it is going to reduce my production costs from $100 a kilogram to $50 a kilogram. And like that attitude is something that we don't have enough of in bioengineering. And I think that tools like BIOS are going to be for people who have that attitude, and they're going to make science a lot more accessible to them. So that's what I'm excited about.
That's amazing. Maybe let's finish the intros, and then I want to hand it off to some of the other scientists on this call. I'm Akash, the head of AI at Bio. I sort of have this cross-functional background between biology and engineering, AI, and so it's just a really fun time to be working on AI scientists, AI for science. Chiara, do you want to go next?
Yes, happy to. Thank you. Hi, everyone. I am Chiara. I am part of the scientific team at Bio, and I also have a background in science and research. I did my PhD in molecular biology, and my main focus was on genomics applied to biomedicine. And I will pass it on to Lucas.
Happy to. One of the scientists in the AI team as well. My background is I'm actually a medical doctor, a physician by training, but also did an under PhD in AI ethic, and like whether or not like all the ethical questions of how much training do you need as a medical doctor to understand what is going on with AI when you deal with patients, stuff like that. So happy to be here. Marco.
Cool. Yeah, I'm Marco. I'm part of the AI team as an engineer. And yeah, my background is in general with engineering, got a master's degree in software engineering. And yeah, I mean, very excited to hear what our main users here, scientists, got to say. So yeah, happy to collect feedback, and for anyone in the chat, if they want to report some bugs, now is also totally fine time. Like I'm here, and the rest of the devs are also in the chat. So yeah.
So maybe I can just kick things off, bouncing off an idea that you presented, Jake. So this idea of like who is the ideal user and the democratization of tools like BIOS. What is it about the current product as it is that is potentially valuable to lots of different user groups? Is it like just truly the capabilities, and like where do you see maybe the niche, or maybe not niche, but where do you see the potential users that would have the most value out of this?
Yeah. So the mental model that I have for science and for AI science is that there is effectively an infinitely large space of scientific questions and scientific experiments that are valid. There's no end to the amount of science that we could do that could be true. And so because that's the case, we need other non-scientific reasons to be interested in a particular topic or to be working on a particular thing. And so science is not this sort of, it doesn't sort of drive itself, if you like. Like we can choose which scientific topics focus our attention, and it's not the sort of internal logic of science that pushes us towards those topics. It's things that we are interested in. And so the interesting thing about a tool like BIOS to me is that you can really quickly generate science which is valid. You can access this sort of infinitely large space of scientific explorations that are scientifically true, in the sense that they're like informed by the literature, that they follow the kind of like logical structure that science needs to have, that they offer questions that can be empirically answered, and so on. But you can shape that exploration by whatever kind of external values and interests might be very personal to you. And so, for example, in my experiments that I've been doing, I gave BIOS a very unguided question. I said, okay, I want to work in metabolic engineering, make me rich, do whatever it takes. Find a high value molecule, find a microbe that can produce it, give me a metabolic engineering strategy to produce that molecule. And indeed, I got a valid scientific research program for that. Then I did a different version of that, a more guided version, where I put in a bunch of constraints that were simulated like realistic constraints that an actual scientist might have, which is to say, like, oh, I work in the American Midwest and I have access to a lot of dairy products. So I want to do something with dairy. And there's a government program that will give me grants to make the supply chain more resilient. And so choose a molecule that the US government thinks is important to have an onshore supply of this molecule. And also my lab works in yeast and we don't know anything about E. coli. And so choose a strategy that works in yeast and not in E. coli. And so you're narrowing the scope of science, and you're generating things that are not just plausible, but also relevant to a particular user. And there's so many different ways that you could shape the scope of an investigation that way in BIOS, and so many different endpoints that you could arrive at.
You know, that's like it's really interesting to hear that, because I definitely sometimes struggle with trying to be super specific about who are the best users, because like let's take ChatGPT for example. I mean, like, who would you say are the target users for ChatGPT? It doesn't really make sense. And I'm not saying that BIOS has quite the exposure of ChatGPT, but even like scientists, like, do these users have to have PhDs in science, or are they maybe just tangentially interested in science, whether it's for like curiosity or business purposes or whatever? And then can BIOS sort of enhance their particular workflows or guide them towards whatever goals that they may have? Chiara, Lucas, I'm curious, how does that sound for both of you, given your background in science and where you see this potentially going and growing further?
Maybe I can go first. I definitely think that BIOS is a tool that is accessible to the expert and novice alike. As Jake was outlining, one can really have a long prompt and start an investigation with many restrictions. But what I like is that even starting with a simpler prompt, or with a non-complex question, let's say, I like that BIOS is able to guide also people through different thinking paths, which is something that is really part of the scientific discovery. You know, this is the first analysis or the first understanding that I have on this problem, and there are 3 different paths to take from here on. Which one do you think is the most interesting? Which one is the most close to you? And let's keep going. And I think that's very beautiful to be able to interact with BIOS in this way.
And only second that. As I was joking, and you probably know that, I was saying like probably BIOS can now do my bachelor thesis in like, I don't know, 20 iterations or something. So I do think the true power right now is for research, like anything university level, I would guess, but not necessarily PhD. Because I think if you also like think about how AI is accelerating science, it's also very hard to kind of like understand where like it's moving so fast. Even if you think about very like, let's say, early niche topics, even within medicine, you really need to understand pretty quickly what is changing and on a daily basis. And I think you also notice how fast biotechnology can be changing, and then basically everything that you recommend for patients also changes. So I do think, for example, if you envision a student in a certain biomedical field, but he might not be able to fully do a full literature review or synthesis on one specific topic, or what is the newest, or for example, on any disease. So I do think like the first persona definitely is like someone who is a scientist. But I think, especially because we optimize so much for the deep research functionality, and I think this is like completely very good. Like this is really as this would be a co-scientist for you. But I do think in the future we probably also will be able to do it more, or let's say less complex. So for example, a patient could also come and understand like the latest. But if you see some of the outputs, I think it's also it would be quite hard for someone to understand, because like the sheer load of information is just hard to grasp sometimes. And if you do like 17 iterations, like, and you're reading 40 pages of a report, even for us as like trained scientists, it can take long. So I imagine it would take endlessly long for a non-scientific trained person.
I think that like the analogy to ChatGPT and the early days of ChatGPT, I think is really beautiful for BIOS. So if you looked at like in the early days of AI, when AI was still being done mainly by scientists and for scientists, if you asked people who were building those early AI models, like what's the use case? What problem are you solving? They would have given you these like really esoteric, like super technical, like things that were informed by their funding agencies, right? It would have been things like, oh, this is what we need is computer vision for satellites so that the Defense Department can like identify terrorist camps from satellite data, right? And like that was like the use case for computer vision. And then like last year, OpenAI released the actual use cases of ChatGPT, and it's like make a fitness plan, make a recipe plan, plan a vacation for my family. And like that's where actually got, right? Like that's what people actually wanted to use AI for. And so BIOS has a real advantage in that in that evolution, in going from the like scientist focused use cases to find the big actual product at the end of the thing, because you guys you're so close, you're closer to the market, right? Like there are a lot of people on BioProtocol building different biologically inspired products from different backgrounds. And so that means like you're closer to the product iteration process, and you're going to find that like recipe planner version faster, I think, than other AI scientist platforms.
Yeah, I hope so. I think the vast range of creative ideas and paths of exploration in DeSci makes it very unique compared to other bodies of scientific work, whether they're like in or outside academia. Jake, I would love maybe if you could walk through some of the conversations that you've had with BIOS, just to maybe take these abstract ideas and go concrete on them. And I know there's like specific aspects of the UX that you have really enjoyed, and just nerding out about that would be super fun.
Cool. Okay. Yeah, I'm gonna post a link to my chat so that people can sort of explore in detail. Should I share my screen as well?
Yeah, I think that would be great.
Okay, I'll do that. I'll share my screen here.
But for those who are watching, I think that chat should be shareable. So feel free to check it out on your own time.
Yeah. Well. Did that work? Did that link get into the chat where people can see it? I think people can see it. Okay.
Works. I can see it.
Okay.
Please.
Everybody see my screen?
Yeah, probably it's better if you can zoom a bit in. We know that it looks very.
How about now?
That's very good.
Okay. All right. So here. Okay. So I'm gonna sort of walk through two different prompts that I tried. These were these were just me playing around. So any resemblance to a real metabolic engineering project is purely coincidental. My inspiration was Make Me Rich. So I gave it the metabolic engineering version of a Make Me Rich prompt. I want to engineer a microbe to produce a high value compound, identify molecules that can be produced by metabolic engineering. That's what we do in synthetic biology. Prioritize molecules that have an existing market. So I want to make something that I know that I can sell. I don't want a market risk for what I'm going to make. Prioritize molecules that don't already have mature metabolic engineering efforts with less literature and less IP filed. So this is a sort of a light guidance, I guess you could say. I want something that hasn't already been invented yet. And then for whatever you find, propose an organism to produce it. and suggest a metabolic engineering strategy to optimize its production. So this is the kind of like most open-ended prompt that I could think of that would get it to a metabolic engineering strategy that would be plausible. And then so yada yada yada. I let it crank on this for 20 iterations. And what I get is, I think, a pretty plausible pathway. So let me see if I can find. Yeah. Okay. So what it came up with is sandalwood oil. It's a metabolic engineering strategy to get yeast to produce sandalwood oil. It gives me some market data on the sandalwood oil market. It tells me where sandalwood oil is currently made. It's made from natural production. It gives me the price per kilogram of natural sandalwood oil, $500 to $2,500 a kilogram. So that's a pretty good price. That's the kind of price that you're usually looking for when you're in a metabolic engineering target. And it tells me that there's currently no commercial scale producers of this alpha santalol, which is this market. I get a techno-economic analysis. So this is already pretty beautiful. This is not something that I necessarily expected it to come up with, because it's something that we as metabolic engineers often don't do early enough in the process. So the techno-economic analysis is the calculation of how much of this product you actually have to be able to manufacture in order to make money. So it gave itself some plausible techno-economic analysis targets for not just making this molecule, but making enough of it that it could actually make money. And then it came up with a particular metabolic engineering strategy to actually produce this molecule. And so in this case, it gives me the plausible metabolic pathway. It tells me that people have tried to use this metabolic pathway to produce this sandalwood oil, but that they weren't hitting the titers. And then it gives me a scaffolding strategy to improve the performance of the pathway. So essentially what it's suggesting is we've got 5 or 6 different enzymes in this metabolic pathway for producing our sandalwood oil. We need to more efficiently shuttle the molecules in between those enzymes in order to get a faster metabolic rate, and therefore through this pipeline or through this pathway. And so what it suggests doing is engineering the proteins to anchor them to an organelle inside the cell, and in this case, to anchor them to the peroxisome. So it's saying we should take these enzymes in the sandalwood pathway, anchor them all into the peroxisome. That's going to concentrate them, bring them all much closer together so that the metabolic intermediates can be shuttled more quickly between them, and therefore produce a higher yield. So this seems to me plausible as a strategy. Then it does an IP analysis or a freedom to operate analysis, saying, has anybody tried this strategy before? Are we sure that engineering these proteins to anchor them to the peroxisome is a novel thing? It's going to be a metabolic engineering strategy that we can get some IP on, and that is going to have freedom to operate. And then it does a review of the IP literature to establish that that's freedom to operate. Okay, so that's the unguided version, right? So that's BIOS just working on Make Me Rich. And I think it was really interesting to me to see that result. So first of all, because it did hit without prompting a lot, like my needs as a user. It did, like if I was like a grad student, for example, wouldn't do a techno-economic analysis unprompted, right? That is the kind of thing that you do need to guide people towards. It's also interesting to me in the context of an AI as a sort of a probability engine, right? Or AI as a kind of an autocomplete engine, because when this is the Marvel movie version of metabolic engineering. Like this is absolutely like paradigmatic, archetypical version of a metabolic engineering project. So it's using one of the most popular organisms, yeast. It's using like an iconic pathway. The isoprenoid biosynthesis pathway has been the foundation of many metabolic engineering projects in the past, including some of the most famous ones. So like for example, artemisinin, the anti-malarial drug is produced in yeast metabolically using this pathway, and it's like one of the most famous like success stories in metabolic engineering. And it's one of these pathways that has a lot of little branches. And so you can go, like there are like dozens of different things that you can branch off this pathway to make different molecules, right? So it found the like core of metabolic engineering. And so it located the molecule there. And then it said, okay, now I need to optimize the production. And so it found a strategy that is also, I think, a pretty high probability strategy in metabolic engineering, which is this scaffolding strategy. So this is something like I would teach this in my metabolic engineering courses. It's the kind of thing that has worked over and over again in metabolic engineering in different ways, right? So it's not always the peroxisome, but the idea that if you want to accelerate a pathway to make more of a particular product, you should take the enzymes and you should cluster them together so that they can pass metabolites in them more efficiently. Like that's something that has worked many times, right? So it found a popular organism, a popular pathway, a popular strategy, and then it gave a sort of twist on it in the sense that it was looking for a novel version of this scaffolding strategy. So it didn't, right, as far as I know, it didn't use one of the existing scaffolding strategies. It came up with an idea that is at least new to me, which is let's link everything to the peroxisome and scaffold them that way. So that's the unguided version of the research. You get the Marvel movie version of a bioengineering project.
When you said the Marvel movie version, my initial thought was like the sci-fi nature, but you were more talking about the like archetypal way of pursuing some of these.
Yeah, it's the hero's journey, right? It's the win. This is the winning formula, right? Like this is how you do metabolic engineering. Yeah,
for sure.
Maybe I also have a follow-up question for you, Jake, as an expert in synthetic biology. If you had to give a feasibility statement on the result, and if you had to estimate how long it would take for the first results experimentally to be there, and how long to complete this suggested experimental workflow, how long would that take?
Ooh, that's a great question. Hold on, let me share my screen again. Can you see it? Yeah, okay. So it gives an experiment. It gives a test case for this, which is something that I wasn't expecting, but it not only gives a strategy, it gives an experimental design for testing that strategy. And so what it says is we're going to engineer 4 strains of brewer's yeast. We're going to start with strains that we already know are good at producing these molecules in the isoprene pathway. We're going to take a version of that pathway where the molecules are totally unscaffolded, right? We're just going to let them float around in the cytosol. We're going to take a version of that pathway that uses a patented technique. We are going to make a version of that pathway that I don't understand what this means. I would have to dig a little deeper to understand what it's referring to when it says the expression control system. This is something about balancing the levels of the enzymes to optimize the titer. And then it says, and then our new system design where we're going to scaffold these enzymes using the scaffolding technology that I came up with to anchor them. And then we'll do triplicate batch fed fermentations. We're going to run them for 120 hours. And then we're going to do mass spec to measure how much xanthol comes out. So it gives the experiment. It gives the experiment. And looking at that, it wouldn't take that long. Like it's not a massive scale experiment. Like this is not a drug trial or something. It doesn't cost $1 billion. It would this could be done for sort of tens of thousand scale to build these 4 strains. And then you would have you would know if it was working. You would know pretty quickly if this was working. And so this is to me, this is a fascinating, what if we actually did that, right? How much better informed would the model be if we could come back with it and say, okay, yeah, we made those 4 strains, and 3 of them looked pretty good, but your design didn't work that well. Which is, of course, the classic grad student experience. And would it be able to take that data and come up with something much more detailed, much more informed by having just four experiments, right? Four designs, right? It's totally plausible that you could get that data and feed it into BIOS at a reasonable price.
Yeah, I think the lab in the loop with the AI scientists is super critical, so that not only do you can you sort of confirm whether or not the scientist is the AI scientist is its outputs are something that you can trust, but I think even from the engineering perspective, you present the opportunity to improve the AI scientist based off of what's actually happening in the world of atoms. Not as cool as these hypotheses are, they are hypotheses, and you need that physical, real world validation. Maybe not the fully at the clinical trial stage, but you need some confidence that the agent is going in the right direction.
Yeah. But so the thing that got me excited about this, right, is this is the model is asking for real world validation at the grad student level of small scale, benchtop, accessible, reasonably fast experimental design. And this is a very different way to think about AI for bio than most of the ecosystem is thinking about it, right? So most of the big AI for bio companies out there are creating gigantic databases that are all focused around one kind of data, right? They're doing things I want to do RNA sequencing on every cell line that was ever made, testing every drug that was ever made, get the this mammoth RNA sequencing database, and then use that to build a model that can predict everything about RNA sequencing, right? And it's this $100 million scale data set that they want to fit into this gigantic model. But what does it look like for these tiny data sets? These little, right? What if the model actually worked like a lab scientist, right? Like most scientists in history have not generated $100 million data sets and then predicted everything about everything, right? Like biology works through with these like touching nature in these tiny little bites, right? And how informative would it actually be to be like, okay, you asked for 4 experiments, you get literally 4 experiments, and that's all you get. Could you learn anything from that, right? Because human scientists do.
I'm curious on like the other end of the spectrum, Jake. So I believe you were running this on autonomous mode on BIOS. There's also within deep research, we have steering mode, which is like 1 iteration, and then we have smart mode, which is like 3 to 5 iterations. If we think about this grad student analogy, how does it compare these different modes to like the real world exercise of working with grad students who are definitely extremely smart, but like depending on their background and experience might need more or less direction at different points in the process?
Let's see. I think the rate of feedback and the shape of the feedback is going to be a really important part of these models, and getting the product side right. I think if you want to make the analogy to the human scientist, I think most scientists will tell you that the relationship that you had with your mentor, how frequently you have meetings with them, the kind of feedback that they give you, like how deeply they dive. Like, are they reading your lab notebook every day, or do you give them a PowerPoint presentation every 2 months? And like that, how you sort of scope that feedback cycle and how you package that feedback for the human user, right? For the human advisor, it definitely does set a lot of the it has a big effect on the direction of the research. And I think it like the biggest effect that it has, I think, is how much do the preferences of the advisor, how deeply are they reflected in the ultimate project, right? So it's like what I want as a scientist is that I want like I want to feel like my preferences are being reflected in what's coming back. Like I think I want my taste to be guiding this model. And so like the level of like the time that I want feedback is I want the model to be like, okay, I found something interesting. Look at this. And you get that aha moment, like you do in human science.
Jake, did you have any other conversations you wanted to share? If not, maybe Chiara or Lucas, if there are any maybe interesting things that you have come across with BIOS, I think it could be cool for the chat to see potentially.
I do have one more to share. If I don't know,
yes, I'm curious to see the
Rich.
This is the more directed version.
Yes, please, Jake.
Okay. All right, here's a link to the chat if anybody wants to dig in more detail. And we're sharing our screen and we're zooming in. All right. So this is a prompt that is simulating a hypothetical human user that is like situated in a particular place and time and has access to particular opportunities, right? So okay, I want to make a high value compound. I have a grant for supply chain resilience. And so find something that's industrially important in the US, but has a fragile supply chain. And I work near a dairy farm. And so find me something that can be made with the waste stream from a dairy farm. And also my lab does yeast engineering. So give me yeast and also find something that hasn't been invented yet. And so this one, I took a more guided approach. I made more choices. Mm-hmm. I think the like interesting moment in guiding this model was when it asked me, when it came back with the first proposed target molecule. And so it said, it gave me what is it? Anthocyanin? Astaxanthin. Astaxanthin is the proposed target molecule. So that was like, that already, like, this was new to me. Where is it? Where's astaxanthin?
Was this steering mode or smart mode?
This was smart mode.
Okay. So
smart mode.
So did it take to get to a molecule? 2, 3, or do you know?
I want to say 2. It came back pretty early. And this was a molecule that I hadn't heard of. It's something that I wasn't aware of, right? So already it's being a little bit more esoteric. It's finding something that's a little bit more niche. Mm-hmm. It's giving me indie movie. It's not giving me Marvel movie. So I had to look up what astaxanthin is. And it turns out it's an antioxidant. It's derived from beta carotene. So it's basically like a super vitamin A. It's like it's a particularly powerful antioxidant. So it's used in wellness products. It's used in products where people, like supplements, where you would want to put an antioxidant. And also is also kind of has the funny niche application that it's a natural pigment. And so like when you buy farmed salmon, this is the thing that they add to the farmed salmon to make it look pink. And so like beautiful metabolic engineering target, totally weird thing that you didn't know that we needed biology to make. Oh yeah, of course, the thing that makes salmon look pink, right? Sure, right? Like let's make that. Let's get rich making that. And so, but the moment that I had was like, I thought, I asked for a supply chain resilience molecule, right? Like what I was expecting was like military, like, whatever explosive manufacturing supply chain, or like the rubber that they need to put in fighter jet tires. Or something that where there was like an obvious need for supply chain resilience. I was like, well, we don't, like if salmon are less pink, it's probably not necessarily going to be in the sort of like of dire strategic loss to the United States military. But it convinced me. That this actually was going to be a useful target to get grant funding around supply chain resilience, because it said here, like, here's a precedent. There was an MIT lab that got $600,000 to produce carotenoids from milk underneath this biorefining and biomanufacturing program. So it convinced me, right? So, and I let it cook. So this is like comparable to the experience that a PI might have with a grad student. The grad student comes up with an idea. The PI is like, no, that's a bad idea, but the PI is wrong. The grad student brings the receipts and keeps going. And so I let it cook. And basically what it came up with was a kind of a 2-part strategy. for making this work in yeast. So the first thing that it has to do is it has to solve the acid toxicity problem of whey. So this is the lactic acid in dairy waste is a big problem. So if you want to process dairy waste, you need microbes that can tolerate a lot of acid. And so it says, okay, we're going to use this acid toxic response transcription factor, and we're going to use this lactate transporter to pump acid out of the cell, and that's how we're going to get acid tolerance. And then to actually make the molecule, it finds two enzymes that are required to make this molecule, CRTW and CRTZ. And it decides that the most important thing for getting a high yield is having a correct balance of these two enzymes so that you can get a sort of consistent conversion across that pathway without accumulating. Like if you have too much of the first enzyme, then it accumulates in the back. And if you have too much of the second enzyme, then it accumulates in the front. So it wants to balance these two enzymes, and it proposes some pathways to keep them balanced, like, for example, like fusing them together or using promoters of the same strength to get them to get them to be balanced, right? And so this is in this case what it comes away with is a strategy that's more niche, that's more useful in a particular context. It gives me the things that I need from a metabolic engineering project, like the techno-economic analysis, and then it drives to a kind of a proof of concept experiment, saying like, make these 4 strains, test them out in acid whey, and then measure the amount of astaxanthin that comes out, and see if you are see if you're productive enough, see if you're hitting the titer that you need in order to make this be an actual commercially viable pathway.
So were you it got your thumbs up also on this one, or was there something that you would have wanted more in this more directed version?
I think the things that are often on my mind are the things that go wrong in metabolic engineering projects are that you didn't do the techno-economic analysis enough, so you end up producing the molecule but not enough to be commercially viable. The freedom to operate analysis can be really tricky. And so you want to make sure you can find enzymes that other people haven't used and develop sort of novel technologies that are protectable. This is an interesting, like maybe like a future state of AI, right? Which is like, oh, it says use CRTZ and CRTW. Well, maybe those are patented, but you can use AI protein engineering to just make new versions of those. If they're patented, right? And so like thinking about that freedom to operate around a project like this is interesting. And then I think the last step is the scalability. Does it work in the actual conditions? And this is a common problem in metabolic engineering projects, is that you get a strain that looks really good in the laboratory, but when you take it to the fucking dairy farm with the dirty toxic feedstock that you write in, and it behaves totally differently. And these are like, to an extent, like I don't know that an AI agent can necessarily correct for these problems, because like these are things that we fucked up as scientists. Like the literature is not a good guide in this sense, that you have a lot of literature that is downplaying that is neglecting techno-economic analysis, that is downplaying the importance of scalability, that is not concerned with IP and freedom to operate. And so these things are like, they're not in the science necessarily, and so you have to find sort of other forms of guidance to get a project that is scientifically valid and also meets these product needs.
That's really interesting. I mean, like, I'm just thinking, like, both points that you just brought up on, like, almost like the supply chain for whether it's manufacturing or just real world use. Thinking through like how we can make the system more robust to like provide like higher baseline levels of performance for these outputs. But yeah, there's definitely like there's definitely an edge that experts have because they have seen like how does this how does this not work in real life that currently the AI scientists really wouldn't have experience with unless somehow it's in a training set on the internet. But yeah, a lot of these edge cases I doubt are well documented.
Yeah, I think like often the edge that the scientists have that is like they know the places where the literature is systematically exaggerates, right? Like they know the stuff that like doesn't really work that well, but that didn't get written into the paper.
So I'm wondering if we can take the last few minutes for Q&A. If anyone in the chat has any questions that we can try to respond to as a group. Let's see. It's a pretty active chat, but I'm trying to see, has anyone seen any questions?
I think we answered most of them on the go. I think we're actually good.
Oh, I see some dairy waste people in the chat. Shout out,
yes.
Shout out to my dairy waste people. Love you guys.
Lucas, maybe
go ahead, Sarah. So Jake, would you suggest to your students to work with BIOS at this point, to run some prompt and come to you with the best ideas to move forward?
Yes, I do. I mean, I think it's right. It's so like the way that you learn is by teaching, right? Like you give your students you put them in supervising positions as early as you can, because it's how they, it's how you develop taste as a scientist. It's how you this like higher level skill set of how do you direct a project to a place that is novel and interesting and solves a real need and is more than just valid science. And so, yeah, absolutely, right. Like this is a great way to just because it keeps you on rails, right? It keeps you on rails. It's not gonna like it will. It's of course like it's not gonna be right about everything, but it keeps you from going off on total flights of fancy that are totally disconnected from anything that is scientifically plausible. Which we do that sometimes.
Can be fun, right?
Anything else, Lucas? Were there any like questions that would be maybe worth just bringing up from the chat, just for those that might be watching this later?
Yeah, I actually think one comment was really good because it was about our let's say the available literature or the literature our current agent can query, because it's primarily English only or effectively English only. And I think this is also something that we definitely have on the roadmap, but I think it's well spotted that this is also a problem of, let's say, the broader AI and LLM ecosystem, especially as everyone is using, most of the projects are actually using the same tools that are available based on open access literature. So if it wasn't much of a question, but it was a good statement and something that I want to emphasize that we will be working on.
Yeah, I think the open access literature is what BIOS has access to right now. Obviously, proprietary like literature behind paywalls would be potentially really valuable to increase literature coverage. Kind of similar on that in that light. If there for any users or prospective users of BIOS, if you have your own like data sets that haven't been published, if you're you can consider using BIOS to like help with analysis there, just so that it can add a second pair of eyes and like maybe validate any like pre-existing analysis that you've had. But yeah, that I think in many ways, so Jake, like a lot of what you showed was like heavily on the literature capabilities of BIOS, but we should definitely mention that the data analysis capabilities of BIOS are quite strong at state-of-the-art levels, according to one of the leading benchmarks. So yeah, there's a ton that you can do with BIOS that I'm excited to see how everyone plays with it. Is there anything else? And otherwise we can probably close here. Okay, well, this was a lot of fun. I really enjoyed this. Thanks, Jake, for giving us the detailed rundown of how you've been playing around with BIOS, and thanks to everyone in the chat for tuning in. And if you have any questions, feel free to send us a DM on X, and we're also down to provide free credits for anyone that has been playing around with the platform. So just let us know, and then we'll get you some credits.
Thank you, everyone. Thanks, Jake.
Thanks, everyone.
Thanks, everybody.
Bye-bye.
Bye bye.